Hi, good morning. I'm your teacher for today, Mrs. Roselle Janicio. So for today, I'm going to discuss to you the different parts of the microscope. So that would be our topic or lesson for today. So we will talk about the microscope. So what are microscopes? So it is used to magnify objects that makes objects look bigger. And it also helps scientists study objects and living things too small to see with our naked eyes. So just like the bacteria, the different organisms or microorganisms that we can see in our hands. So we cannot see anything there. There is no dirt. Okay, cannot see the microorganisms because they are very small. So for example, we have here a needle. Okay, if you would just look at the needle or the sewing needle, you can say that it is clean. But then when we use a microscope to magnify the microorganisms, if there are microorganisms that is on the tip of the on the tip of the needle, this is what we can see. So since microorganisms are very small, we cannot see those organisms by just using our naked eye. So we need to use an equipment or tool to see those microorganisms, and that equipment is the microscope. So microscope helps us to magnify the small microorganisms or the microorganisms that cannot be seen using our naked eye. So as what you can see there, this is the tip of sewing needle, which is covered in bacteria. So if we're going to use a microscope, then we can see that there are different microorganisms, even if it looks clean for us. We just use our naked eye. And for today, we will discuss about different parts of the microscope. So why do we need to know the different parts of the microscope? So that we will know how to use it or manipulate the microscope. If we do not know the parts of the microscope, then eventually we cannot use it properly. We cannot manipulate the microscope properly. So we need to learn first the different parts of the microscope. So here are the different parts of the microscope. So we have the ocular lens or the eyepiece, then the body tube, the revolving nose piece, the arm of the microscope. It's just like our arms, but it's different, of course. We also have the objectives. We also have the stage clips, the stage, coarse and fine adjustment knob, the diaphragm, the light source, and the base. So before we go through or discuss the different function of each part, I'll just introduce to you the different types of microscopes. As what we all know, there are different types of microscope, and these are the simple microscope, which has only one lens. The compound microscope has two sets of lenses, and it can magnify things 100 to 200 times larger than they really are. So these are the microscope that we use in our school, that we have in our science laboratory. And of course, the electron microscope, which is considered as the most powerful type of microscope that can magnify objects up to 300,000 times. So they do not use lenses, but they use electrons to enlarge the image. That's why we call it as an electron microscope. If you are not familiar with electron, it is one of the subatomic particle of atoms. Electron, protons, and neutrons. So electrons. So they use this type of microscope uses electrons to magnify to enlarge or to magnify the objects or the microorganisms rather. So we have the simple microscope. It only uses one lens. That is the reason we call it a simple microscope, because it has a very simple structure. It's just like that. So this is Logan's simple microscope, circa 1871. And of course the compound microscope. So this is the microscope that we usually see in our science laboratory, or any that we usually use whenever we have an experiment that we need to use a microscope to magnify microorganisms. And of course the electron microscope, so it's bigger than the two types of microscope. So that's it. We have 3 types of microscope. So going back, we will just give the function or description of each part of the microscope. So we have here the ocular lens or the eyepiece. This is the lens of the microscope that you look through. So just like in binoculars, there is 2 eyepieces in binoculars where you will use to see the objects or to magnify objects. And in this case, in the case of the microscope, we use the ocular lens or the eyepiece to magnify the different organisms that you want to see under the microscope. Second would be the parts adjustment knob. This is a knob wherein you will turn it just like this. The large knob on the microscope, because there are 2 knobs, the larger one, the bigger one, and the smaller one. And the larger one, that would be the coarse adjustment knob. So that this is the knob that you turn to bring the object into focus. So before we can magnify the microorganism, we can actually see the clear version or clear image of the microorganism. We need to use first the coarse adjustment knob. If it is clear enough, then we can use the fine adjustment knob. So where is the fine adjustment knob? There it is. It is the smaller knob on the microscope that brings the image into focus. So it makes the image clearer. Next, we have the arm. It is the part of the microscope supporting the body tube, and it is where you will hold the compound microscope. We also have the body tube, the part of the microscope that holds the eyepiece and the objective lenses. As what you can see, it connects the eyepiece and then the objective lenses. We also have the nosepiece. The nosepiece is the part of the bottom of the body tube that holds the objective lenses and allows them to be turned. So you can actually turn and rotate the nosepiece depending on what objective you will use. We also have the different objective lens. So when you turn the nosepiece, you can actually use, if you will turn the nosepiece if you want to use a specific type of objective lens. So we have different objective lens. We have the HPO or the high power objective lens. So the lens that magnifies the object the greatest amount. That is the reason we call it as HPO or high power objective lens. So it usually magnifies up to 40 times, 40x. LPO or low power objective lens, the lens that magnifies the object the least amount. So usually used to find the object magnifies only 3x or 4x. So usually we we do not call it as low power objective. We call it as scanner. Okay, scanner. And we have the middle power objective lens. This is the type of objective that I call as LPO or low power objective. Okay, so it's different from the scanner. Scanner, you'll use that if the specimen is macroscopic, meaning it's small, but you can actually see it using your naked eye. So you will start with the scanner. But if you cannot really, or you cannot see anything, then you can proceed to LPO or the middle power objective lens or the low power objective. So this is the lens that usually magnifies the objective more than the scanner lens but less than the high power lens. So it is usually 10x to 20x. And we also have the stage. This is the flat part below the objective lens where the slide is placed. So the slide is where the specimen are fixed, where you will put the specimen. Then that slide, you will put it on the stage. So it's where the slide is placed so that you can see the microorganisms. And to fix it, you will use the clip, stage clip. So the part that holds the slide in place so it doesn't move. And the diaphragm, it is the part that controls the amount of light entering the field of view. So you need to check first the diaphragm of the microscope. If you need a very large amount of light, then you can just adjust the diaphragm. You can choose the bigger or larger opening of the diaphragm so that you can have a larger or high amount of light source. And of course, the light source. This is the lamp. So there is what we call electric compound microscope, wherein you will use electricity for you to use the microscope, and your light source is a lamp. But if it is not electric compound microscope, then you can just use the mirror, wherein you need to find the light source, maybe fluorescent light or natural light will do, under the stage that sends light through the object being viewed. And of course, we have the base. This is the bottom part that supports the rest of the microscope. By the way, class, it's important for you to know that whenever we are handling the compound microscope, we need to hold it in its arm and its base so that it is handled with care, because compound microscopes or microscopes generally are very expensive. So always carry microscope with one hand holding the arm and one hand under the base. So this is the proper way of handling or holding the microscope. And for the field of view, it is the area or circle when you look at through the eyepiece. It is the area that you see when looking through the eyepiece. This is how it looks like when you look at the microscope. So there is circular field of view, and then the rest would be black. And if we're going to compare the power of magnification of different objectives, so you have the HPO, LPO, and scanner. We have the high power objective lens. We have the LPO or low power objective lens. And we also have the scanner. So we can see better details with higher power of magnification. But we cannot see as much of the image. Example, 7x compared it to 10x. So the image is magnified better in 10x. So which of this image will be viewed at a higher power of magnification? If it is magnified or zoomed in, then that would be on the right image. It is viewed at a higher power of magnification. And we have here the different objectives. So they are identified with the different colors. For this scanner's color red, 4x, magnification of 4x. For low power objective, we have 10x or 10. High power objective, 40. And of course, the oil immersion objective, wherein you need to use a special oil to be put on the specimen so that you can magnify the object up to 100x. So it's a very powerful objective. Has a very high power of magnification. The higher the power of magnification is, the more magnified the object or the microorganism or the specimen will be. So what's my power? So if you want to calculate the power of magnification, you will just multiply the power of the ocular lens or the eyepiece, because the eyepiece that we are using to view the specimen also have magnification power. You can see it on the side part of the eyepiece. So in this case, we have 10x. And the power objective or the objective lens that we are using is 40x. So how do you get the power of magnification? You just multiply it. So 40 times 10 is equal to 400x. So that would be the power of magnification. Okay. And for that, so before we end our discussion, I would like you to, if you want the video, if you like the video, please like this video and of course subscribe. Thank you. Bye.